real-time array. Actually, we are now using the real-time array, and in the real-time array, we have the 96 well plate. Eight wells are for the controls, and the other 88 wells either printed by single gene that we want to assess, and we use the plate for 88 patients, or it is printed with 88 genes that we need to assess, or a pathway, and we use it for a single patient. We have again control and tested cells or tissue. We isolate the RNA or the microRNA, and then we do the RT-PCR and the real-time PCR. I will speak about the microRNA. We have a large panel of microRNA tested. And these are the differentially expressed microRNA in hepatocellular carcinoma. We have here the microRNA tested and the target genes that the microRNA either stimulate or inhibit, and the function or the molecular function that this microRNA is involved in. So, for example, microRNA 15 and microRNA 16 are working on, their target is the BCL2, and therefore they affect apoptosis, whereas microRNA 100-06A, for example, is working through the RB gene, and it affects the cancer pathogenesis. This study of that has been done on our lab in 2012. We assessed microRNA and metastasis-related genes in Egyptian breast cancer patients. The genes that were assessed include the matrix metalloprotease and the vascular endothelial growth factor and cyclin G1. As we can see in this figure, we can show that there is a dramatic decrease in the microRNA in tumor tissue compared to normal tissue. So breast cancer patients show an upregulation in certain microRNAs and in the matrix metalloprotease genes, and they show downregulation in other microRNAs in cancer tissue compared to adjacent non-neoplastic tissue, and this was statistically significant. And from this study, we end by a panel of microRNAs, microRNA 10, 21, 155, 373, 330B, 126, 17B, 335, that could be used to differentiate between metastatic and non-metastatic breast cancer patient on serology. And this is how is data presented using the real-time microarray. And this, at the left side, we can see the genes that were the microRNAs that were differentially expressed between in hepatocellular carcinoma. And we can also see that in this study we assessed microRNAs in chronic hepatitis, cirrhosis, and hepatocellular carcinoma, and we found significant changes between groups regarding the microRNA present. So, for example, microRNA 885-5P, there is the expression of this microRNA differs significantly between the three groups. So it can differentiate between chronic hepatitis, cirrhosis, and hepatocellular carcinoma, whereas if we look for microRNA sample 222, it can only differentiate cirrhosis from the other groups. So we have work on colorectal cancer. We assessed microRNAs in adenoma and normal and inflammatory bowel disease, and in colorectal carcinoma. And again, we can see that there is differential expression of the microRNAs between the groups. So we can see that group 1, group 2, and group 3. Group 3 is the hepatocellular carcinoma, group 2 is the adenoma, group 1 is the inflammatory bowel disease, and the control group. And we can see that there is a change through the cascade from the normal control or the adenoma to carcinoma. So what's completely green in the control group turned into red and green in different sides. Then we had a study on 157 female patients with locally recurrent inoperable metastatic breast cancer, and in this study, we looked for biomarkers in the EGFR platelet-derived growth factor pathway that can tell us who will be the responders and who will be the refractory, together with examining CYP2G6 polymorphism. And we found that 56 genes were upregulated and 34 were downregulated, and among these, only JAK, the one in red were upregulated and the one in green were downregulated, were used in a panel that we have further used it in a validation set, and it proved that this panel can differentiate between patients who will respond to tamoxifen therapy and those who will not respond. This is how we present data on the real-time array. This is the volcano plot and the genes that make the panel. And then we have molecular changes in EGFR pathway. This is our final part of the presentation. Molecular changes in EGFR pathway and RAS-RAF genes in metastatic and non-metastatic colorectal cancer. In this study, we have assessed a large number of patients, 396 colorectal cancer patients, 240 were metastatic and 156 were non-metastatic. All were assessed for KRAS BRAF mutation and were assessed this by conventional PCR and line strep assay and immunohistochemistry. We also tested other genes in the EGFR pathway that are related to the KRAS and BRAF, such as the PI3K, MEK, and ERK expression by immunohistochemistry and quantitative real-time PCR. And we have seen that some of these were correlated to prognosis. So our results were that out of the 396 patients who were tested for EGFR, 164 were positive and 232 were negative. We assessed KRAS in EGFR negative patient because this means something related to treatment, and we found that 120 were positive, 112 were negative. Then we started to look whether there is an interplay or a cooperation between the genes in this cascade or not, and actually we found that the PI3K was correlated with all other genes in the cascade except for the BRAF and KRAS, and we found that also the KRAS or the PI3K was the only gene correlated with EGFR in the non-metastatic, whereas in the metastatic cascade, the PI3K correlated with other genes. What I need to say is that we found a great difference between the metastatic and non-metastatic regarding the expression of the EGFR-related gene, but EGFR pathway-related genes. And by this, I will end my talk. And next generation sequencing, as I told you, we have done only 10 cases, and we hope that by the next meeting of the NCI, we can have data to share with you. Thank you.
Thank you, Dr. Habib, for this detailed presentation. We will have 5 minutes for Dr. Moni if you have any questions or comments. Any question? Okay. We will take like a stretch break, maybe for